Thank you very much. So my name is Bree Allen. I'm the president of business unit within Rigaku Corporation, which focuses on spectroscopy and handheld analytical devices. And today I'll be talking about a new generation of handheld spectrometer for chemical identification. So thank you very much to the WCO for this opportunity to make this presentation. So just very briefly, in a point of introduction, Rigaku Corporation is a Japanese-based company based in Tokyo. It's a privately held company, almost 70 years old. And its specialty is in X-ray analytical equipment, primarily large laboratory equipment for very precise measurements for material science and material characterization. Rigaku Analytical Devices is a business unit based in the US, in the Boston area, which again is specialized in handheld ruggedized analytical technology. So taking the laboratory systems and miniaturizing them into a very portable form, which is also very capable for field portable analysis. So Rigaku's mission is to contribute to the enhancement of humanity through scientific and technological development. So that's what we focus on, is developing analytical technology, furthering analytical technology to solve problems that our customers are experiencing. And that's what I'll be talking about today. So I'll be focusing on bringing analytical technology into the field. And this is critical for customs agencies, of course, for importation inspection, for looking for smuggling of illicit products, or for doing quality control and validation that products are exactly what they are specified to be. But the applications go beyond that. The applications go to threat diagnosis, threat detection, looking for not only things like narcotics, but also explosives and even chemical warfare agents in the military theater. Some of our customers include a very nice list of references, including Japan Customs is one of our largest customers worldwide. So we've deployed our spectrometers to Japan Customs in multiple sites, again, for the detection of illicit materials like narcotics. But we also have systems in use in the US Customs and Border Patrol, throughout Europe, in several different European customs agencies, including France, Belgium, Finland, and then also in Asia Pacific, including Philippines and others. So I'll give an introduction here to our flagship product, which is called the ResQ product. And this is a handheld device which can do chemical identification across a very wide range of different material types. So the objective here is to have a device that you can hold in your hand, and within less than a minute, you can get confirmatory identification of what an unknown material is, be it an illicit material or be it a perfectly benign material. So for those of you that are not familiar with this technique, our system is based around Raman spectroscopy. The Raman spectroscopy is a very old, well understood technique in which you are exciting a sample with a laser, and then through molecular vibration excitation, the material itself will release energy, and we gather that energy and transform it into a spectrum. And that's what you see there in the middle of the slide. That spectral signature is unique to the material which is creating that spectrum. So it's almost like a fingerprint of the material. And that can be a fingerprint of a pure material, or it can be a fingerprint of a mixture. So obviously, if you're testing unknown samples, you never know if they're a pure material or if they're a mixture of combined materials. That spectral signature is unique to that combination, to whatever that substance is. So we gather that information, and then by deconvoluting that spectrum and then comparing it against a library within the system, then we can do confident identification of the material. That's how Raman spectroscopy works in a very basic form. So Raman spectrometers have been around for years and years. In laboratory form, they've been around for going on 30 years. In portable handheld form, they've been around the industry for 10 to 15 years in widespread use. The nice thing about Raman is that it has high specificity. So you can do quite a bit of identification no matter what organic material type that you're testing, be it narcotics, chemical weapons, explosives, toxic chemicals, pesticides, even pharmaceuticals, steroids. It has a wide range of different applications. One of the biggest advantages of Raman technology is that you can scan through packaging material. So as a customs official, you do not need to open up the material and expose the inspector to the substance which you're testing. You can test directly through any material that is really translucent or can pass the laser energy through it. It's very easy to use. So the systems nowadays are designed to be point and shoot. So very little training and very little, very short training time is needed to actually operate these systems effectively. There's no consumables, so there's no sample preparation, there's no sample digestion, and it is a very close to non-destructive technique. So you can actually test samples over and over again with no sample degradation at all. In terms of workflow, again, it's very simple, and we've built these systems to be very user-intuitive. On our system here, you would simply push the button, scan, and then within 1 minute or less, sometimes even just 30 seconds, you will then get a result screen. And that's based on the spectrum that you're receiving from the sample after excitation. So you can get a very clear result screen, which will tell you and indicate to you, is this a threat? For example, in the top case, this is TNT, which is an explosive material, or down below, an acrolein, which is a non-threat or a benign material. It can identify for you. As a third option, our system actually will tell you if it's an illicit narcotic as well, and we'll label that a bright yellow if it's a fentanyl or heroin or cocaine. But again, you can get these results very quickly. And they're very informative. So you get the NIOSH information as well as the hazard diamond, and then you also have access to spectral information as well. So the key to these systems is, as you can probably guess, the size of the libraries. So if you have a handheld Raman system that has a very small library, then obviously your scope of capability is going to be very limited and very narrow. So to have the most effective instrument possible, we've had to invest a lot of money and a lot of time in developing and generating very large libraries. So our system has a library consisting of more than 12,500 chemicals, different pure compounds in it. And our system can also deconvolve mixtures so that it can identify mixtures of a product. So if you have up to five different components in a mixture, it will identify each of those 5 different components. Included in this very large library is, of course, target materials, which you are most concerned with, including illicit drugs, including cutting agents and precursor materials, explosive materials, toxic chemicals and toxic materials, also chemical weapons, and pesticides. One of our big initiatives within Rigaku is to continually invest in developing and growing this library. So we can provide our customers and our users with the most capable instrument possible. So we spend a lot of time generating new spectra from new compounds. When you think about narcotics in particular, tens to hundreds of new narcotics are being produced and developed and synthesized every week. So we, as a producer and manufacturer of these types of devices, must try to do whatever we can to keep up with that and add those compounds into our library. And we do try to do that. And then once we generate those libraries and we expand those libraries, then we deploy them to all of our customers immediately. And our customers can update their instruments over email very easily. So that's one aspect of our business which we must apply continuous focus on. The other nice thing about this system is that you as a customer can generate your own libraries. So in particular, if you're in Eastern Europe or you're in China and you see synthesized new narcotics being developed, you can add those to the library very easily in your laboratory. And then with a push of a button, you can deploy those added spectra to your fleet of instruments very, very quickly. So that means you can also, as an organization, you can keep up with new materials that your inspectors are finding as well. Our instrument not only looks for chemical identification and alerts the inspector to the presence of threats, but what it will also do is monitor results over time and look for combinations of materials that could indicate the manufacturing or the development of threats. So what that means is if a user or an inspector is inspecting multiple packages, they may find 2 or 3 different chemicals which are benign and are not threats individually. However, the presence of those materials together can indicate that someone is making an improvised explosive or making a narcotic or even making a chemical warfare agent. So our system not only has that library of pure chemicals in it, it also has a library of recipes. And again, it will monitor those results to indicate that, in this example, it found citric acid and hexamine, which by themselves are not terribly threatening, but in combination could indicate that someone is making HMTD, which is a highly energetic explosive. So in terms of Raman, and I assume that many of you are familiar with Raman technology over the years. Again, the Raman advantages are you can scan through packaging. It ignores water content, so you don't have to worry about drying samples or pre-concentrating samples. The Raman spectroscopy technique, water is invisible to it, so that doesn't affect the sampling at all. And you don't have to contact or interact with the sample in any way. But with traditional Raman systems, they had certain limitations that were due to the characteristics of the sample. So they were very, very good for white powders. They were very good for clear liquids. But as soon as you started testing organically derived materials, as soon as you started testing colored materials, meaning cut with pigments or cut with colored cutting agents, or you were testing samples through colored packaging. Maybe you have a red plastic bag or you have a green bottle. Those traditional Raman systems produced what's called fluorescence. And what that means in layman's terms is a large amount of noise in the spectrum, which prohibited the instrument from being able to make a confident identification. So for those of you that have used Raman over the years, you know this, and this is a big pain point and a big limitation of Raman systems. And so this is just more information about fluorescence. Traditional Raman systems, which had this fluorescence problem, were based around 785 nanometer lasers. So this is the characteristic wavelength of the laser, which is exciting the sample. 785 nanometer laser systems were used because 785 nanometer lasers are very prevalent and very easy to get and very cheap. They're used in the telecom industry for monitoring telecom communications. So they're very easy to get and very easy to integrate into a handheld system. But they had these use case limitations where, again, if you tested colored samples, you would have problems getting identification. So what we've done is we've gone to a different laser source, a 1064 nanometer laser source, which is a longer wavelength laser. Unfortunately, it is more expensive and it's a little bit harder to engineer. But the benefit is that you get much, much more incremental capability. You can do those white powders, you can do the clear liquids, but then in addition, you can do the identification of colored material and you can test through color packaging. So it gives you a much broader range of application capabilities. And I'll talk in much more detail about that in a minute. Here on the left side, in the blue spectra, you see this presence of heavy fluorescence. So this is a typical spectrum from a 785 nanometer laser system, when you're testing a colored material or an organically derived material, you get this very high presence of fluorescence. And you can try to continue testing for a long period of time to try to reduce some of that fluorescence, but ultimately you reach an asymptote where you cannot eliminate all of that fluorescence. With 1064, which you see on the right side, you don't get that fluorescence problem. You get a very nice, clear, very concise spectrum. And that gives you a very high confidence in your identification. So that's what we're looking for, and that's what we're after. So again, 1064 systems can do that colored, the white powders and the clear liquids, but it can also do the colored materials. From personal experience, I know working with Customs and Border Patrol along the southern border, there are smugglers that are actually bringing methamphetamine into the US, and because they know the limitations of the Raman systems, they're actually diluting the methamphetamine in a liquid form. They're diluting it with diesel fuel. And diesel fuel has a very high fluorescence for 785 nanometer systems. So they know that by diluting the methamphetamine with diesel, they're masking it, and they're prohibiting or restricting the ability of the Raman systems from being able to identify the methamphetamine. But with 1064, you can measure that diesel methamphetamine mixture, and within a few seconds, you get that resolved identification. It will actually tell you diesel fuel and methamphetamine. So you can see the benefit is immediate, especially when you're dealing with colored material. And if you think about improvised explosives or homemade explosives, nowadays those are never going to be made with pure laboratory quality chemicals. They're going to be made with household chemicals and things you can buy at a drugstore, which are typically going to be discolored materials. So the ability to measure colored material becomes all the more critical as we think about homemade explosives. So I'll give you some examples of applications where the 1064 laser helps quite a bit. Thank you. So this is an example of a study we did with the Edgewood Chemical and Biological Center, part of the US Army, looking at chemical warfare agents. So you see on the left side an example of sarin, pure sarin, and then on the right side, pure sarin, but in a munitions grade or degraded form. And you can see using the 785 nanometer laser versus the 1064, the difference that you get with the spectrum. And quite a bit of difference there. So with 785, you're unable to do a confident identification, but with 1064, you're able to get nice crisp peaks, which enable you to get much more confident identification. For explosives and precursors, you can see from the examples we're showing here how many of those raw ingredients that go into homemade explosives can be colored material, including citric acid, including hexamine, ammonium nitrate, and that's where the 1064 can help you quite a bit. So here are some spectral comparisons, TATP, HMTD, and R-salt. Again, 785 versus 1064. Again, the key point here is with 1064, you're getting a very nice, high-quality, crisp spectrum from which you can do your identification. And with 785, it's just not possible. For narcotics maybe present the largest challenge for colored material analysis, because the narcotics can be cut with literally anything, and in many cases are obviously not pure. So here's an example of heroin, and with 785, in order to actually measure street quality heroin, in a lot of cases, you need to use a technique called SIRS, which is surface enhanced Raman spectroscopy. With 1064, SIRS is not necessary. The problem with SIRS is that there's consumable cost and that you also have to interact with the sample. Again, with 1064, no sample interaction. You can test it directly and get an immediate answer. Skip ahead here. Here's an example of some material that was smuggled in from Mexico into the US, or they tried to smuggle it in. And this is cocaine wrapped in plastic, several layers of plastic, and then a layer of what's called sriracha, or hot sauce, and then more layers of plastic. And with 785, they were not able to do the identification because of the presence of the sriracha hot sauce, which is a strong red pigment. With 1064, you can get an immediate analysis. You can see that this is an example of a test report from our system. And included with the test report are images of the sample itself. Our system actually has an integrated camera for that type of sample imaging. So that gets saved directly with the result and then can be exported immediately over Wi-Fi. And here's an example of a body carry of methamphetamine and vegetable oil mixture. So this was carried on a woman's body, trying to be smuggled across the Mexico-US border. And you can see the green color there and the layers of plastic. And again, this was analysis directly through that masking material. Synthetic cannabinoids are becoming more and more of a problem, especially in the US. And cannabinoids are highly fluorescent material. So again, with 1064, or you need the 1064 laser in order to get confident identification of cannabinoids. An example of our continual development on our libraries is the fentanyl variants. So up until recently, there were maybe 5 different variants of fentanyl that were worrisome in the US, including fentanyl, including carfentanil, and some other variants. This year, we're up to about 15 to 20 different variants of fentanyl. And I'm sure next year, that will have doubled again. So one thing that we have to continually invest in is expanding that library to include those variants. And that's what we're showing here, is that we continually develop and deploy those new chemical signatures through our systems. So I'm running out of time, so just in summary, the 1064 Raman, very highly ruggedized, very compact system. It was introduced in 2015 and is currently being deployed all over the world. And it provides a fluorescent-free signal, so you can do much more incremental chemical identification than with traditional Raman systems. So we have the Rigaku ResQ systems just outside the door here, and we'd be very, very happy to give a demonstration or answer any questions for you, or give a demonstration to you there. So any questions for me now? Yes, sir. Coming. Yes.
And is there a logbook in the system that can check? Because you know, an officer can be tempted and to forget his responsibility.
Yes.
So my question is, how is that connectable into equipment, and how to make sure the message can get in another system?
Very good question. Thank you. So to answer your first question, yes, the system immediately after the conclusion of the test generates a PDF of the report. And that PDF report includes the analysis results. It includes the spectra. It includes any sample images you took, as well as any qualitative information. Let's say you have inspector number or inspector name or sample number. That is all included, saved in memory, but then also included in the report. And then you can email that off immediately. Sure, yep. The memory of the system is 100% locked down. So you as a customer cannot delete any readings from the memory. This is 21 CFR compliant, secure data results. So you cannot tamper with the results and you cannot delete the results. They are forever on that instrument.
Maybe one more question.
Sure.
How ruggedized is your equipment? If it falls down, how strong is it in terms of rain, shocks,
Yes. Environment. So it was designed for the U.S. military, around U.S. military specifications. So it is drop proof and vibration proof and shock-proof. So it's been thoroughly tested. In terms of sealing, it's rated at IP68, which means you can take the system and dunk it. And many of our customers actually need to be able to dunk the system in order to decontaminate it. So you can take a dilute bleach solution and take the system and dunk it under water, and it's fully sealed. You're welcome. Thank you. Any other questions? Okay, thank you very much.